everyone. So, tau is an axonal protein that has slightly different variants between mice and humans. And what we found during my summer research with Dr. Bergold is that when you introduce a human variant into a mouse brain, this leads to better memory performance after brain injury. So the brain is like a giant electrical circuit, and you can think of axons as the wires that keep it cohesive, as seen up there on the top. And tau is a protein that binds to structural elements within the axon and keeps it stable. You might have heard of tau. It's a pretty famous protein, commonly studied in its relation to Alzheimer's. But we are specifically interested in its role in traumatic brain injury. Now, the brain is quite squishy. It's a lot like Jell-O. So when there's trauma, there's acceleration and deacceleration, which can lead to injury much further beneath the surface, like with white matter. So across studies, white matter has been shown to be critical for traumatic brain injury, and the accumulation of tau is also seen. So what we wanted to do is study this, but the variation between the two proteins poses a question. And a big problem in research is being able to extend human data towards basic science research. So what we did is use a humanized tau mouse to look at the effects of brain injury and memory performance later. So we induced the brain injury on normal mice and then the humanized mice. And we tested their memory performance on a task called active place avoidance. So this is really hard. The mouse is placed in a rotating arena and based on visual cues in the room, has to avoid getting shocked. And we tested the mice over 5 trials, and these are their movement graphs in the 5th trial. So on top is the normal mouse. You can see that it still is entering the shock zone. That's each of the red dots, and is getting shocked repeatedly. It has not really understood where the shock zone is. So impaired memory performance after brain injury. In contrast, the humanized mouse has much fewer shocks. On the 5th trial, it is able to remember where the shock zone is, demonstrating that it has improved memory performance after brain injury. So this was done in the acute time point, 2 weeks after injury. And all we can infer is that human tau might have some protective role in the short term. However, when we test these mice 6 months after injury, the picture is not so clear. They demonstrate similar performance to the normal mice. So what remains to be done is to repeat our experiments, confirm our results, and try and understand really what human tau is doing. Are the wires stronger? Where's the long-term damage and why? And how can we use this for patients? Thank you.